Welcome to Pharmacy to Dose, the Critical Care Podcast. I'm your host, Nick Peters. Wherever you are and whoever you are listening, thank you. Okay, fun episode today. You may or may not have known that the FDA Cellular Tissue and Gene Therapies Advisory Committee had a meeting last Friday, November 22nd, to discuss a supplemental biologics license application. That's the only description on the YouTube video recording itself, or if you are watching the live stream. You'd never know this application that they were discussing is actually Andexanet Alfa. And that's how much I love you, friends. I watched this 6-hour webinar not once, but twice. So you may notice this is a rare solo pod, no guest. So almost every single thing I'm going to be saying are going to be quotes or references to quotes from some of the committee members and subject matter experts. It was the FDA. And I mentioned the Cellular Tissue and Gene Therapies, or the CTGT Advisory Committee. And then, of course, the AstraZeneca, or what they referred to in the meeting as the sponsor. So there was also an open public hearing where those in the public, mainly physicians, couple pharmacists, one lay person, they got 4 minutes to argue their case as to why Andexanet Alfa should or should not get approved. I definitely encourage everyone to, if you're interested, watch this on YouTube or, follow along. So in full transparency, one of the things, so we're going to kind of, if you're watching on the YouTube, great, love that. This would be a great one to do that for, because there will be screenshots of the slides that they were talking about. And on the actual slide that I'll be sharing, it's got a timestamp of when that was taken from the video for full transparency. So if you're like, wait a second, when were they talking about this? You could go exactly to the time on there. Now, you might have to move forward a couple seconds here or there, right? Because they might have talked about something different at the start of that slide or something like that. But that is there. Other big point, trying to discuss facts and talking through some of this information. I want to be clear. I don't necessarily feel super strongly one way or the other. What I want to do is want to make sure that that we are using safe and effective medications. And some of these experts brought in really great points, both for and possibly against the side of this. And I think it's interesting ideas, a lot of which maybe we haven't necessarily talked about. But with that being said, I think we probably need to start off with, what was the purpose of the meeting, right? So they said this in kind of different parts throughout, but ultimately it kind of came down to 5 separate things. So discuss and obtain input on the benefit risk profile of Andexanet Alfa, which is indicated for the treatment of patients treated with rivaroxaban or apixaban when reversal of anticoagulation is needed due to life-threatening or uncontrolled bleeding. This product was granted accelerated approval based on the surrogate endpoint in the decrease in anti-factor 10A activity in healthy volunteers. We're also here to discuss the increased risk of thrombosis observed with Andexanet Alfa for the proposed indication in the context of clinical efficacy seen in Annexa I. We're asking the committee, right, the FDA, and the FDA is asking the Cellular Tissue and Gene Therapies Advisory Committee to opine on whether the data that has been submitted in this application and presented to the committee constitutes clinical benefit. And the questions are purely on the clinical data submitted in the application. So the FDA reminded the committee that the converting of an accelerated approval to a traditional approval requires demonstration of a clinical benefit. And this next part was repeated throughout. The way the FDA defines clinical benefit is a measure of how a patient feels, functions, and survives. It is the benchmark, and this is the requirement, and it applies to every product that we approve. So that's the basis of what they're looking to do and talk through, right? Talk through the accelerated approval to go to a full traditional approval. So let's go ahead and start kind of following along. We're going to kind of have a little slideshow. Don't worry, pod listeners. This is gonna feel like a old hat, like we're going through stuff. But if you want to follow along, definitely come to the YouTube channel. So there were two main discussion topics um, that was the focus of this. So, discuss whether the treatment effect on the study's primary efficacy endpoint constitutes a clinical benefit to patients. And they talked about three specific things. Does the effect on hematoma volume change at 12 hours alone constitute clinical benefit? Should neurologic status at 24 hours, MRS at day 30, and overall mortality be incorporated into the assessment of benefit of Andexanet? And does anti-factor 10A reduction have a role in the assessment of the benefit of Andexanet alpha? And the second pieces are the serious risks of Andexanet as demonstrated in an XI acceptable in the indicated population and in the context of the clinical efficacy demonstrated in an XI, right? If anyone's been following along to episodes we've had, it's the balance, right? Of it did show benefit in that hemostatic efficacy, but the increase in thrombotic events. But we're going to get into all of that. This was actually really, really fascinating. I had never seen the kind of how behind the scenes this is. And it was very um, illuminating. So let's again remember the proposed indication. This is all patients with life-threatening or uncontrolled bleeding. We focused a lot in the discussion. We'll focus on ICH, but remember this accelerated approval to traditional approval would be for any life-threatening or uncontrolled bleeding anticoagulation reversal for rivaroxaban or apixaban. So why are we here, right? So the discussion centered around these trials. The far left, the Annexa A and the Annexa R, the first two studies, they were healthy volunteers showing rapid reversal of the factor 10A inhibitor activity. It was how they got the accelerated approval. The Annexa 4 arm was a kind of single arm study that showed hemostatic benefit, and this evidence supported the accelerated approval, but uh, the that wasn't enough data itself from the FDA's perspective. So when they look for the evidentiary standard for approval from an accelerated approval perspective, which is what they had, improvement over available therapy and the effect on the surrogate endpoint or intermediate clinical endpoint is reasonably likely to predict clinical benefit. So remember, does anti-factor 10A reduction actually predict a reduction in hematoma volume expansion? So this is from the FDA accelerated approval letter. So the new data from bleeding patients they're referencing the Annexa 4 study, indicated that changes in anti-factor 10A activity do not necessarily predict the clinical outcomes, while the risk of thrombosis in these patients was elevated. Therefore, FDA recommended that they go ahead with an RCT to evaluate the clinical benefit versus the standard of care. So why did they mandate, right, the Annexa I study? There were a few things issued by the FDA. There were key design factors that went into play with this, right? 440 patients, at least 440 adult patients, verify the hemostatic efficacy in the acute ICH, include assessments of NIHSS and CT-MRI at 12 hours post-randomization, 30-day safety follow-up, and then an adjudication committee to determine hemostatic efficacy. So a couple things here, and what I'll do kind of throughout this is in a 6-hour period, right, they'll bounce around and things. So what I try to do is try to summarize things like that. So I'll be bringing in pieces from other parts of the webinar, but there was a question of who chose the indication to be studied. Was it by the FDA or by the sponsor? And we negotiated with the FDA and chose the ICH population because you can verify hemostatic efficacy in a very precise way with brain imaging, and you can't quantify a change in bleeding for GI bleeding and such. They mentioned it's not a feasible study to design a study for all types of bleeding, yet this is what the proposed approval would be for. So one study in a specific population was designed to evaluate that surrogate endpoint and evaluate this population with intracranial bleeds. So let's talk through the timeline for study design feedback that was presented. So first patient enrolled. They had a protocol amendment in April 2020. And then this is where things get a little bit interesting. So in August and October of 2020, FDA review division consulted with the Division of Neurology Products, and they decided that the 24-hour endpoint could be meaningful in ICH, but the most important measures of neurologic functional assessment should be later, such as 90 days. So the sponsor, this is a quote from the director of regulatory affairs at AstraZeneca. The sponsor considered this feedback, but decided to continue with the original study design aligned to the 2018 PMR for hemostatic efficacy. So they're saying what the initial study design, how that is sufficient here. They added exploratory endpoints of neurologic assessments at 24 hours, but the length of study follow-up continued to be through that 30-day period. Now, it is interesting to note. We're going to kind of definitely try to show as much of a fair balance of both sides or both sides of the facts as possible. And what the folks from AstraZeneca noted was the average enrollment rate was 1 patient per center per year. So that actually, I hadn't thought about that, and I'm sure anyone in research was like, duh. But essentially, the that was one of the kind of reasons that they gave as to why they kind of thought that. So um, AstraZeneca, then the first piece of the presentation was AstraZeneca, and then it went to the FDA. They had an open public hearing and then kind of committee discussion. That was kind of the breakdown of it. And AstraZeneca had speakers to highlight why this is an unmet need, detail the efficacy and safety of Andexanet Alfa, give clinical perspective, and there was a moderator for the Q&A from the committee. And a key point here mentioned by the first speaker is the time to anticoagulation reversal, right? The goal is less than 30 minutes, reverse the anticoagulant. It's even highlighted in an FDA slide that the mean time to treatment post randomization was 25 minutes in the Andexanet arm, and usual care arm was 30 minutes. But I think it's notable that we should point out a piece from the Annexa-I protocol that I think is pretty illuminating here. So to allow for the equal timing of assessments in both treatment arms, Andexanet must be prepared prior to randomization for all patients, irrespective of the ultimate randomization outcome. Now, I find this funny because one of the things that AstraZeneca actually mentioned was that Andexanet Alfa could be given even earlier outside of the research setting. I do get that. I understand that enrollment um, and randomization takes time. However, I also know that ordering, making, bringing this drug takes longer than I think that the data from this Annexa-I study shows, because this agent was pre-prepared, and that is just not, there's no way that people are doing that. Now, the next slide is our first mention of VTE prophylaxis and how emergency medicine and critical care teams are equipped, how we can manage acute VTE. Actually, when I was listening, it stood out to me so much. I wanted to actually write down all of the quotes in the beginning piece that mentioned this. So much confidence in our team. So there are 5 of them. So the first one, while thrombotic events can occur, critical care teams are well equipped to manage the risk in the ICU setting. Emergency and critical care teams are fully equipped to manage these complications. Protocols are in place to identify and treat thrombotic events. While the risk of thrombosis may be elevated, critical care teams are well equipped to monitor and manage these complications. And we can manage the thrombotic risk by following guideline recommended approaches to VTE prophylaxis and restarting anticoagulation therapy. So there was a pretty illuminating slide that we have seen half of before. So there's one slide that shows those who received anticoagulation and the patients who had thrombotic events, right? And it's about 5% in both arms. But when they look to the other side, patients who did not receive anticoagulation or it was started after the first thromboembolic event, uh, 23% of patients in the Andexanet Alfa arm, 7.7% in the usual care arm. Now, the frustrating thing is, this is the slide that, or the information that we've seen, right, was that the thromboembolic rates who received greater than one dose and 70% of patients received a dose, right? And knowing that other information, that kind of startling information was available, or maybe it wasn't, and they just got it available. It seems like it probably would be if you have the other side. Feels a little frustrating, but it's really good that we got to see it here. Now, it was discussed later that a lot of these parenteral anticoagulant doses are prophylactic doses. And to quote the patient safety physician, restarting therapeutic anticoagulation usually occurs much later. And uh, one thing that was fairly notable, they focus on some parenteral anticoagulation, but in the first 7 days, no patients received reinitiation of oral anticoagulation. We're going to put a pin in that and come back to it just a second here. Now, another thing that was highlighted was looking at the thromboembolic rate in patients with no hemostatic treatment and compared to PCC. And those with no treatment had about a 12% thrombotic rate, similar to the Andexanet Alfa group, right? And PCC or other, however, was just under 5%, 4.8%. So a pretty big difference from that perspective. Now, they detail the multiple approaches to re-anticoagulation after ICH. And you'll see this reference of Gorman 2023. So they're referencing a 15-year retrospective study describing different approaches for patients with a VTE post-ICH. And they talk about no anticoagulation, subtherapeutic, stepwise approach, or going to immediate therapeutic dose. And they found that the recurrence or progression of VTE was less in those who had delayed escalation. But one thing that I think is important to note was that the mean interval from ICH admission to therapeutic anticoagulation in this 15-year period that AstraZeneca was referencing was 21 and a half days, 19 to 26 days. So that feels important when we talk about the earlier timing of some of these things. So AstraZeneca, they highlighted some differences in baseline characteristics from the high and low dose Andexanet Alfa treatment arms. And what you can see here, it's kind of highlighted that the high dose had a higher incidence of cardiac failure, little bit higher diastolic volume, higher hematoma volume. And we're certainly implying that this was the reason for the difference in the thrombotic rate, some of those differences in baseline characteristics. And so they wanted to highlight from the single arm Anexa 4 study. They wanted to use that data to demonstrate that the high dose was safe, kind of comparing not only the hemostatic efficacy, but mortality and thrombotic events being similar there. So one of the questions was, and this was kind of a point, was talking about the dosing. Have we figured that out? Should the high dose still be used in things? And a question came up that was, have you looked at doing alternate regimens? And they ran a demonstration, essentially like a PK simulation, that they were giving a low dose to all high dose eligible patients. And what they found was that you won't achieve hemostasis in this model But their definition of hemostasis was achieving the anti-factor 10A target of 30 nanograms per ml. So what they're saying is you gave the high dose, you didn't get that full target all the way down. Now, notable. So, what I didn't know about this before we kind of get into this quickly, I didn't realize today I learned when you're applying, you submit all your data to the FDA and they rerun all the statistics, they re-adjudicate some of these things. So, they essentially run all their own stats and things by themselves, which makes complete sense. Um, but the FDA showed this slide towards the end showing that the high dose Andexanet Alfa arm actually had a higher rate of thromboembolism compared to the low dose arm and a higher mortality. So now one could argue they took it more recently. Are they going to be a higher bleed risk? That's not necessarily what was being told to us in the earlier, us being the audience or the FDA and the committee members. That wasn't how it was being discussed or brought up. It was brought up that they were similar, and the result that they create similar outcomes. And this would argue not, right? When we look at the thromboembolism for the high versus low, almost 21% versus 13%. And the mortality, almost 40% in the high dose to 25% in the low dose group. So pretty stark differences in that. So the treatment emergent adverse effects kind of look similar effects, a little over 80%. But one of the key things was diving into the treatment emergent serious adverse effects. So infections and nervous system disorders due to the underlying bleeding event were the most common. These are an expectation in a critically unwell population. That's the quote from the patient safety physician. But the difference in thrombotic events is notable for both myocardial infarction and acute ischemic stroke. 5% versus 0.8% for ischemic stroke. And then myocardial infarction, 3% versus just about 0.5%. Now, the AstraZeneca highlighted that there was a difference in the number of FDA assessed and those that were independently adjudicated thrombotic rates. And comparing the FDA, the usual care thromboembolic rate went from 5.6 to 6.9%. And the Andexanet Alfa thromboembolic rate went from 10.9% to 14.6%. But it's notable how this slide ended. We will focus on these adjudicated events for the remainder of the safety presentation and likely thinking, hope we forget about the increase from 10.9 to 14.6%. Now the FDA kind of broke down their thromboembolic events, right, and showing this a similar thing of ischemic stroke and myocardial infarction being the two most common. Now this is just a graph kind of showing the difference, right, of 7.8% difference usual care between Andexanet Alfa and those thrombotic events. But one of the key pieces of information that was presented was the number of days to the thromboembolic event, and a median of 3.5 days in the Andexanet arm versus 16 days in the usual care arm. Now, even more in the first 3 days, half of the patients, right, who had a thromboembolic event was in the first 3 days. We just saw a slide that says the median time to therapeutic anticoagulation was 21.5 days. And over 50% had it in the first 3 days, compared to a usual care arm of it was one event, right? 6% had it in the first 3 days. And another very visual effect, if you're not watching the YouTube, you can go to social media and I'll have this posted, it's a graph and it shows the patients that had thrombotic events and death. And it starts off, it has the Andexanet Alfa patients on top and usual care patients on the bottom. And it visually shows when thrombotic events and deaths happen day-wise in relation to restarting anticoagulation. And The more dots to the left mean it happened closer to your kind of sentinel event. And there's a kind of looks like a swimming race is getting started near the top with that kind of median 3 days seen in the Andexanet Alpha group. When the FDA kind of went into their discussion on the incidence of thromboembolic events in those who restarted versus those who did not, that's similar information to what we were talking about. The the safety takeaway slide from AstraZeneca is that there's a higher rate of thrombotic events with Andexanet, and it's a known risk when restoring physiologic coagulation in patients with an underlying thrombotic risk who are also bleeding. A proposed USPI update informing use in specific patients with high baseline thrombotic risk. Now, the FDA safety discussion mentions that the thromboembolic rate was double in the Andexanet Alpha arm compared to the usual care arm. They occurred much earlier, and nearly half of them occurred in patients who were on VTE prophylaxis. So the idea that VTE prophylaxis is going to save our day, that was what everyone told us over and over and over and over and over again in this. Nearly half of them occurred in patients who were on them. And the deaths from the thromboembolic events were higher in the Andexanet Alpha arm compared with usual care. Now, one thing that I wanted to point out that I really like is thinking about groups probably should not receive Andexanet Alpha. Maybe we inform our use in those patients with high baseline thrombotic risk. And the idea is those with heart failure or a history of stroke or MI, maybe they should not receive this. And that feels like a smart proposal, again, with the idea that this could be approved for a big population. So let's shift to the efficacy side of things. And I just want to point out that the FDA did not review data from edoxaban and enoxaparin patients in this application. So some of the first slides that we saw were figure S2 from the Annexa supplement that shows that Andexanet Alpha rapidly reduced those anti-factor 10A activity, that it reduced it to a 94% reduction at 2 hours versus 27% in the usual care arm. We've definitely seen that. Now, this next information is key. So when simply comparing anti-factor 10A level reduction, Andexanet Alpha was superior to usual care. But even Dr. Ashkan Shwamanesh, the Annexa investigator, noted anti-factor 10A is a good, reasonable predictor of the anticoagulant activity. I don't think it's the only marker, and I don't think it should be used in isolation. So you see the anti-factor 10A activity reduction, right? 95% in Andexanet, almost 30% in the usual care. But an important point is that there was no difference in this reduction between non-responders and responders. So what does this mean? This means that if you received Andexanet Alpha, your anti-factor 10A activity was reduced 95%. But that did not predict if you were going to respond to your treatment or not respond to your treatment. And similarly, in the usual care arm, the anti-factor 10A activity change was 28% in the responders and 32% in those non-responders. So one of the questions that was posed is, do you need 95% reduction? That's a great question. Now, one of my favorite comparisons from this meeting was from, I believe it was Dr. Thomas Wu. Anti-factor 10A activity being used as a biomarker, I think of like this. Low pH is bad in DKA, but if you fix the pH with sodium bicarbonate, it doesn't help the patients. So is there actually a correlation with anti-factor 10A levels with hemostatic efficacy? That was a pretty stark comparison. Now, I'm sure most people would predict this was coming, that AstraZeneca was prepared, and they presented a multivariate analysis showing that the reduction from baseline to nadir of anti-factor 10A activity did show a benefit for hemostatic efficacy when you control for a lot of other factors. So the relationship between anti-factor 10A activity and hematoma expansions pointed out its multifactorial complicated process. So when you adjust for competing risk factors, so the things here: baseline anti-factor 10A activity, baseline hematoma volume, baseline diastolic blood pressure, and time from symptom onset to treatment, it was presented that this reduction did show a significant difference. Now, the FDA is quick to point out that this data wasn't shared with them. They haven't looked at it or anything. Now, it was kind of discussed back and forth. This slide obviously made appearances throughout. It was discussed back and forth that this was a subgroup defined after randomizations. Take this with a grain of salt. And the AstraZeneca response, we recognize that this is a subgroup defined after randomization, and those patients who responded and achieved hemostatic efficacy in the usual care arm are potentially a healthier group of patients whose bleed stopped without specific reversal. So these type of responder analyses may be inherently biased against Indexa. Again, this is the whole. They got accelerated approval for these anti-factor 10A activity levels, and when we talk about them, these levels are now inherently biased. Now, it's also noted another huge problem with the idea of using anti-factor 10 activity is that it is not widely available for rapid testing. Around the clock testing, they noted in the presentation, is only available for about 20% of patients. And even in the research study environment, they didn't mandate these levels for everybody, only in those who presented greater than after 15 hours, or if there was unknown time of last anticoagulant dose ingested. So one of the open public hearings says there's a balance of having availability of lab tests and delaying treatments for 30 to 60 minutes. So interesting point for the anti-factor 10A activity. So definitely should point out, especially even using the FDA analysis, that the primary composite outcome of hemostatic efficacy and they focus on hematoma volume. Remember, it's a composite of three different things: hematoma volume less than or equal to 35% increase, an IHSS increase of less than or equal to 7 from baseline, and no rescue therapy received. So if we remember, Andexanet Alfa met that primary outcome, a difference of about 12%. So the chart is mixed up if you're on the YouTube channel, but what they're showing is when splitting up Europe and the US populations, it actually appears that the Europeans were more likely to have an improvement in the hemostatic efficacy compared to the US. Now, there's a lot of discussion on the 12-hour time point, and some of the mentions was that the FDA supposedly picked this 12-hour time point, but in an attempt to show the 24-hour exploratory outcomes, they show that there was an improvement in NIHSS at 24 hours here. So as we shift from that primary outcome to MRS and mortality, an interesting finding is that those with a baseline MRS of 0 to 3 had a higher rate of MRS of 4 to 6 at day 30. And this was both in the Andexanet Alfa arm and the usual care arm. So not unique to one. And this even maintained when you were breaking out responders and non-responders. However, it maintained for Andexanet Alfa that those with a MRS baseline of 0 to 3 maintained or had a higher percent. It went from 59% had an MRS of 4 to 6 at day 30. So it was 68 in the full group, 59% in the responders, whereas in the usual care arm, it was a MRS in those who had a baseline of 0 to 3, that 30-day 4 to 6 MRS score, 61% in the usual care, but when this broke out to responders, it was only 41%. But there was no statistically significant difference in that overall 30-day mortality. And one of the things that was pointed out is kind of comparing a number needed to treat for not achieving hemostatic efficacy versus the number needed to harm for thromboembolic events. And specifically focusing on kind of those long-term neutral outcomes, AstraZeneca said to fully elucidate, we'd need a study of 3,000 patients We didn't have the appropriate length of follow-up to establish it within a trial. Okay. So let's summarize these efficacy points. So the study primary efficacy endpoint of the composite hemostatic efficacy was met, 66% in the AA arm versus 53% in the usual care arm. The largest treatment effect was seen in the change of hematoma volume. It was notable that hematoma volume measurements past 12 hours were not submitted. There was some discussion that they had some samples between 12 to 72 hours. I think it was 79. There were like 3 events, but it's like, how are those chosen? It feels very picked. I don't understand. But I think that was to show that how little events happened in hours 12 to 72, and that the hour 12 was the key difference there. So, the secondary endpoint was the anti-factor 10A activity. And although the AA arm had a big change in reduction, there was no correlation between anti-factor 10A reduction and hemostatic efficacy, meaning that anti-factor 10A activity reduction didn't predict clinical benefit. And the outcomes of those additional efficacy endpoints, like neurologic deterioration at 24 hours, overall mortality, and 30-day worse MRS score, were similar between those AA and usual care arms. And then, they highlighted the kind of those differences in the MRS scores between them. So let's dive into the discussion here. And the discussion question number one, essentially, discuss whether the treatment effect on the study's primary endpoint constitutes a clinical benefit to patients. I think one of the big things is and that was asked over and over, how do we define clinical benefit? I want to remind you, right? The FDA defines clinical benefits, a measure of how a patient feels, functions, and survives. So one of the points discussed, better hemostasis leads to better outcomes, but this wasn't necessarily evident in the Annexa I numbers. And this was a really, really good question, I thought. Is there some advantage to Andexanet Alfa beyond what was being measured in this trial? And do we think that the markers for clinical outcome aren't sensitive enough to detect that positive change? It was also noted that there's no alternative to Andexanet Alfa, and this complicates matters. Now, a lot of physicians seem to share this thought, right? The need for immediate and effective hemostasis, and that outweighs any potential downstream risk of thrombosis. That was a direct quote. Now, a clinical benefit is a secondary outcome for Annexa I with hematoma volume change and hemostatic efficacy being the primary outcome. So, was the trial designed or meant to measure these sorts of outcomes? I actually thought this was a really great way to sum it up. Is the goal to assess the drug as the reversal for DOACs? And we probably shouldn't necessarily look at long-term outcomes. If we are looking at how the drug is treating ICH and other complex disease processes, yes, we need to look at all of the long-term outcomes. I thought that was a really, really well said way of breaking that down. Now, discussion question 2, are the serious risks of Andexanet Alfa as demonstrated in XI acceptable in the indicated population and in the context of clinical efficacy? So how do we define the indicated population? So this was a really well said statement, kind of thinking about the indicated population. So I wish I had a better understanding to whether long-term measurements like death and neurologic functioning are not important to the physicians that are going to use this. But I haven't heard someone clearly articulate why this wouldn't matter. So they talked about over and over that the clinicians, I agree, you see it, it's so polarizing. Clinicians are desperate to use this drug. And it's the first time that the committee members noted that they've seen so many clinicians in the open hearing asking, pleading. They were like begging for it is honestly what it felt like when you were listening to it. Now, this was a very key question. If we're looking at all sorts of life-threatening or uncontrolled bleeding, should we be looking at MRS? Is that not inherently associated with just ICH? But remember, this was the FDA's response earlier. Those metrics are associated with the population that was studied in the trial that is submitted for us to verify the clinical benefit for this indication. So, what they're saying, this is what they used in the Andexanet study, and this is what they submitted for us to review. So, these are the metrics that we need to review. They had a layperson on the committee, and he had a pretty stark comparison. And I hope that's one of those that doesn't actually come true, but he was saying it reminds me of opioids. You know, when the indication of opioids was to eliminate pain, and it was clear that it did that. But unfortunately, we were then left with 2 decades of side effects. And I get fearful that in an Andexanet Alfa, they mentioned 88% of these patients are on this agent for AFib, on it for stroke prevention. Are we exposing a very, very large population to the use of a drug that may, in fact, damage or harm them? So, applying this data to different populations is a very important question. Um, you know, the in the discussion, the members kind of talked about how the only real data that we can look at to evaluate the serious risks across all populations is mortality. That's the only one that is representative of the entire proposed indicated population. And, you know, this is very specific data for a very specific population being used to approve a drug for a much larger indication, a much larger population, right? You know, the question was brought up, are we going to have a lot of GI bleeds being given this drug? What's that potential consequence? We have no way of knowing that looking at this data, right? And one of the speakers in the open public hearing was Paul Dobesh. And in full transparency, Paul Dobesh is a pro Andexanet Alpha pharmacist clinician. We've been working, he was going to come on the podcast, but with the committee hearing coming up, we wanted to delay. I'm sure I'm going to be able to get Paul to come on and talk through some of this stuff. And his study had a decent amount of GI bleeding patients, but it is a large study, but it's prospective or it's retrospective database kind of observational. And if we're holding these new drugs and guidelines to gold standard RCT standards, shouldn't we be having the same for drugs that are getting approved? Right? That's a question that I'm asking. And how do we weigh the MRS knowing that it doesn't relate to that entire proposed population? So, one of the things that I think is important is to talk about some of the populations that we've studied this in that we know and have some data on. And the two that instantly come to mind are populations that get anticoagulation reversed a lot. And we have evidence that this drug might not have the best outcomes in those patients. So first is the Annexa S study. Now, specifically, what the Annexa S study is, it was a study that was looking at the use of Andexanet Alfa for surgery. So it was looking for patients that needed urgent surgical reversal, and they enrolled 10 patients ultimately. That's all before it got withdrawn. They had an efficacy set, meaning they included 6 patients that had a baseline anti-factor 10A activity of 75 nanograms per minute. And the way this protocol worked, the drug was started at incision and continued through the surgery. No data on how long it was continued or anything, but out of those 6 patients, 5 of them died. 7 out of the 10 patients overall had serious adverse effects. There was a Annexa RS that was going to be looking at that reversal that was withdrawn due to its single arm nature. So really the only data we have in surgery, because those patients were excluded from the Annexa I, is this data from the Annexa S from clinicaltrials.gov. And the other thing, right, is it's used for cardiac surgery populations. The known effect that Andexanet Alfa has on heparin and the effect of heparin resistance, right? It binds to heparin antithrombin complexes. So think about our patients that are maybe on a Pixaban that need an urgent surgery, right? How is that going to work? So I think there are logistical things when we think about applying this to different populations. Now, when we say the FDA has a chance to do the funniest thing ever, one of the things that was pointed out was, can we get more information with ICH, but maybe have this approved with other populations? And that would be truly tremendous if this was approved for every population but ICH, knowing that the by far amount of data we have is from the Annexa I and ICH patients. So I'm hoping that it is not the case. So some big takeaways here. So I think what everyone isn't necessarily saying is that the anti-factor 10A activity may reflect the drug concentration, but does that actually reflect the drug's anticoagulant activity? I thought one of the hematologists, and it's important to note actually, there was a EM physician. He had actually, I think he was from Baylor, and he had a presentation, and it said for his 4-minute open public hearing, and the following presentation contains graphic content. Was on it, and it showed his anecdotal experiences of a crike gone wrong. But he mentions that on the committee, there are no emergency medicine physicians. And I do think that's an important point as a group of physicians, um, despite them loving the Andexanet Alfa reversal agent, putting that aside, having not a single representative and having a lot of hematologists, a lot of pediatric hematologists and things, I can understand wanting a little bit of that acute care perspective. So I do understand that. But when we were talking about, the thinking about studies and correlation and causality. So one of the things that was pointed out was the majority of these studies establish a correlation between hematoma expansion and worse outcomes. And the effect on hematoma volume is the evidence of target engagement. And a test of the idea is that a change in hematoma volume may improve outcomes, but they didn't necessarily see that. And what makes it even harder and complicates things is that they saw this increase in thrombotic events, which could be potentially nullifying these benefits of affecting hematoma volume. Someone put it actually really eloquently. This is a real regulatory dilemma because this drug met the primary endpoint. The primary endpoint wasn't necessarily designed to evaluate how a person feels, functions, and survives. And I'm not sure, based on that definition, does it seem to provide that clinical benefit? But they noted multiple times that based on clinicians who use this drug is important for other indications and other purposes. So I want to close out with how they summarized the end of the hearing, how they summarized questions 1 and 2. Thinking about question 1 and the efficacy endpoints, we are challenged by a few aspects. It's a drug used acutely, but do we want to look at long-term effects? It is very nuanced in a complex landscape, and there are a lot of comorbidities in this population that are prone to strokes. The thing is most confounding about the data is we do not see a decrease in hematoma volume with reduction in anti-factor 10A levels, but this doesn't connect to a clear clinical outcome that has a benefit. Meanwhile, we have an increase in thromboembolic events and mortality. So what is the goal? Is the goal just an immediate reversal of the anticoagulation, or is it about a long-term impact? It's almost a question of definition. Question 2, is dose part of the equation? When we're thinking about risk, right, and thinking about weighing the risk in the context of the general population, but also in the knowing this Annexa I population, this discussion and data was geared towards ICH, but it's hard to really view or understand the overall risk for the entire indicated population. A lot of things to talk about as dose part of the equation when it comes to risk. It was asked, do we know the right dose? And I think that's the challenge on giving a definitive, clear answer if the risks are acceptable in this indicated population. So The conclusion from the FDA that the decrease in anti-factor 10A did not predict effects on primary endpoint of hemostasis at 12 hours, comparable efficacy, although they met their primary endpoint, thrombosis risk a concern. So we'll end with this. It was a 6-hour hearing, and I was, talking to a few people. And I think in the beginning of it, the I think the thought was that was not necessarily positive from the way questions were being asked, answers, things that were being shown, slides, etc. But I think it's really, really important to note that they brought up multiple times how many clinicians strongly want and need this as they mentioned as a part of their toolbox. And so they supposedly have about a month to make a decision. People have asked me what I think. Um, Honestly, I have no idea. If you want to make me make a prediction, I guess I would say that my guess is it gets approved with some sort of restriction or some sort of follow-up requirement would be my guess based on what they were saying. My true thought is we've been talking, the discussion's been focused so much on ICH and really digging into the nitty-gritty of all of those things. And what my takeaway is the overall intended population is so different from the data that we have. It's indicated for patients treated with rivaroxaban or apixaban. Patients treated with rivaroxaban or apixaban when reversal of anticoagulation is needed due to life-threatening or uncontrolled bleeding. I guess I would just say that my only hesitation, my only pause is we don't necessarily know what's going to happen in some of these other vulnerable populations in a high quality gold standard trial that we're holding our guidelines and other national governing bodies to. Feels like our governing bodies overlooking drugs should probably have somewhat of a similar standard. So that's all my thoughts. I wanted to be sure to get this out and hopefully allow everyone to hear it, take it in. I hope this was helpful. And again, yeah, I'm repeating quotes and things from the webinar and things itself. So the timestamps are on it. Go back and look. The YouTube stream will be in the podcast episode description. It'll be on the social media links. This is getting posted literally right after I record it. So you're gonna be able to listen to this in real time. Please let me know what you think. So the audio is going to be available Wednesday. The video will be Friday. Be remiss not to say, right? It's Wednesday before Thanksgiving. Very thankful for each and every listener, guest, current guest, friend of the pod. Y'all are the best. It's absolutely the best getting to collaborate with so many incredible, amazing, talented, fantastic pharmacists. It's the best thing. And with that, I'm Nick Peters, and this is Pharmacy to Dose, the Critical Care Podcast.